Thank you very much, Dr. Siju Thomas sir. Am I audible? Hello.
Yes, sir. Yes,
Am I
sir. Yes.
Okay. Thank you. Okay, and I wanted to know that whether my screen is visible to you. Is my screen visible to you?
Yes, sir.
Yes.
Okay. Thank you, Dr. Siju Thomas for a very nice, simple introduction, and thank you for organizing such a wonderful webinar. And I'm very happy to see several hopeful faces here, including my students, my colleagues, teachers from other colleges. And thank you very much. But in fact, I feel that I'm talking to my computer screen. Anyway, this is a very different scenario that we are facing during the pandemic. Anyway, we are trying to adjust with the situations. Okay, so today I'm not talking about anything related to taxonomy, but I mean related, yeah, but not taxonomy really. So I have opted a title, Identification of Plants, a Task Beyond Theory. So I mean, actually, taxonomy is visible, or taxonomy, the result of taxonomy has to be evident from the field identification and identifying new taxa or differences in already identified taxa. So I'm trying to give you some insights in identification of plants, and I would like to take you to something which is related to a task beyond theory. Okay, so I was thinking where to start with. So now, why do we consider taxonomy as an important subject, even though when we hear about the title taxonomy, both the teachers as well as students will have a kind of fear factor in their mind. So not only studying, I mean, it is a fear factor even for teachers also. So how it happens and how we can overcome that. So first of all, we have to understand the significance of taxonomy, learning taxonomy, and from here on, I would tell it is identification of plants. So we have about 7,402 angiosperm or flowering plants in the Western Ghats, all along the Western Ghats, and of which about 31% are endemic to the region. And in Kerala, we are very blessed with flora and fauna diversity. We have about 4,968 listed species from Kerala, of which about 34% are endemic, about 1,300. And another important factor is that about 252 species are seen only in Kerala, nowhere else in the world. So they are endemic to Kerala. So let us see who identify plants in Kerala. There are few medicinal practitioners, especially Ayurveda, Unani, and traditional medicinal practitioners, plant collectors for several purposes, even for fuel wood collection. Nowadays, it is very rare, but fuel wood collectors, they used to identify which plant would be better fuel wood. Then tribal people, timber merchants, and old-aged people. And these people can at least identify plants by their local names. And they may have their own local name also. But it might not be valued with the new generation, because from very young stage itself, our kids are constrained within the walls. And they are not even looking outside, because they're always engaged with something or the other inside the house or inside the wall. So then there are about around 100 people who are trained taxonomists in Kerala who can identify between 1,000 to 3,000 species. Then when we go to the upper level, people like Padma Shri Dr. K.S. Manilal, who can identify about more than 80% of the floristic diversity in Kerala. And there are so many, about 10 to 20 people who can identify more than about 80% of the plants which are so far reported from Kerala. And most of them, many of those people are from this Manilal school. Okay, then we can assume that what are the tasks with us. So with that task keeping in mind, let us go ahead. So why we need to identify plants? When we want to relate plant taxonomy or identification, there are so many things coming up. Ecology, plant ecology, anatomy, reproductive botany, plant-animal interaction. If you want to carry out any studies in ecology, anatomy, reproductive botany, plant-animal interaction, so on and so forth, you have to have a thorough understanding in identifying plants in the field. So unless you know identification in the field, means you may not be able to do these interdisciplinary aspect research. Then when you want to collect some raw materials for medicine, for any other purpose, you should know identification of plants. Then when someone is engaged in identifying or extracting new drugs, you require taxonomy, molecular data. To collect molecular data based on species, then you should know taxonomy to some extent. Then plant breeding for new varieties, for new aspects, then again you require crop improvement, then clonal selection, then evolutionary studies. And evolutionary studies include phylogenetics and other molecular aspects. Unless you know where to find a particular taxon, where you get a particular taxon, and how you can distinguish that from a related taxa, closely related taxa, you should know taxonomy. Okay, then what is identification and how you should go about it? So identification process starts from field work. Extensive field work and field conditions are required for identification process, because you have to start from the habitat, the environment, the place in which it is growing, and things like that. Okay, so how you should identify? You have to identify a plant correctly. You have to identify a plant with confidence, and you have to identify a plant with authenticity. Then evidently, you have to have some evidence with which you have identified a particular plant as A, O, B, or C species. Then you become an expert. Okay, so fieldwork is an unavoidable factor for identification of plants. How you can go about it? You may need to use any existing flora or key, or you can go and use online resources. But I strongly advise you go online resources once you are thorough with at least one local flora. Okay, then you can go with experts. Especially when you start your process of field identification or plant identification, even if you are confident in your identification, you have to consult an expert and see whether your identification is correct or not. And if there is something went wrong, you try identify what went wrong, why you have identified it as a different species than the actual one. So for that, I think an expert is very much necessary. Then you can go to different herbaria, and match your specimen with other herbaria deposited in different recognized herbarium. Okay, so what constraint that we have in identifying plants? The high diversity, be it Himalaya or in Kerala, it is, any place if you select, be it any habitat, it is very difficult, because the habitat, the species diversity within a limited area is very, very high. And be it trees, lianas, climbers, shrubs, herbs, or whatever that matter, it is very difficult, because in 1 square meter itself, you get many number of species. Okay, like this, many of us may not have seen these plants, at least some of them are very rare. And some of them might have been available in the online, but several of them are available in the natural habitat. So there are so many plants, so many flowers, so many beauties available around you. And another thing is that not only high diversity and variation, unisexuality and spatial separation of sexes. So, for example, Embellia ribes is a unisexual plant. It may be a monoecious or polygamo-dioecious plant, but if you are getting male plant alone and no characters available with you about the female plant, then you will be in trouble, because in keys there might be characters of both males and females described. So especially when you deal with families such as Euphorbiaceae and Lauraceae, it is very difficult. Okay, so these are some of the main threats or main hurdles that you have to face during the initial stages of taxonomy, or whenever you want to enter into identification of plants. Okay, now let us see what are the difficulties. Whenever you go for plant taxonomy or identification, you feel that things are very technical, okay, but very precise. And the scientific names, it's very difficult to remember and even to pronounce scientific names, some of the names. Then, if the dissimilarity is higher, like we can easily distinguish two related things, then it is okay. But if you, even if you go through keys, or even if you get closely associated taxa, you can see more similarities rather than differences or dissimilarities. That is another difficulty. Then several characters are too confusing. Okay, then you might be dealing with variation. This is not the similarity dissimilarity. This variation might be related to the spatial variation, or climate related variation, or soil related variation. You might have heard of ecophenes or ecotypes. Then remembering scientific names. So some of you might be very what do you say, knowledgeable about a few factors, but unless you are comfortable with all these difficulties, or unless you try to overcome these difficulties, it will be very difficult to nurture a very good genuine interest in identification of plants. Okay, then let us try understand these issues separately. Technical, there are so many technical terms, but you just imagine that if instead of this particular term spatulate, if the descriptions are like broadened tip with a narrow base. So here in this plant, the Scolia species or Plectrantha species, Lamiaceae family, the lower petal is a kind of spatulate shape, spatula shape. Okay, so then there comes so many other characters like corona, pollinia. I'm just selecting few technical terms, pollinia, things like that. So even if you know these terms, you have to have practical knowledge on how to separate out or how to identify, because some of the minute characters on corolla or pollinia has to be checked when you need to identify certain plants with minute characters. Okay, this is a pollinia of an orchid. Then I mean Asclepiadaceae also you get pollinia. Then another technical term is gynostigium. I just wanted to bring you some technical terms. Okay, so if you don't know, or if you do not have a thorough understanding about these technical terms, and I will deal with a few other technical terms also from each and every part. So I mean, unless you are thorough with these technical part, you still have the difficulty in handling technical. Okay, so how can we overcome this? There are a few sites of which I would recommend this vplants glossary site, plant glossary site. I mean, this is a wonderful site, and they are updating the technical details, new technical terms, and also the ones which are not included in the glossary. They are updating it very nicely, and they are explaining each and every characters, and also they may have explanations of technical terms within the explanation, or there are plates or pictures like this. They have very good explanatory pictures or drawings. And another thing, another wonderful picture for higher level learning of identification and taxonomy is Taxonomy of Vascular Plants by George H.M. Lawrence. This is a wonderful book, and if you are thorough with the technical terms and how to go about this particular book or using this book, then you might not feel difficulty in handling flora or any keys. Then how to remember these technical terms? Then I would like to tell you is that a better practice is to draw characters. I mean, nowadays it's very difficult even to ask students to draw diagrams. I mean, I don't tell you that we need to become artists or we need to have very wonderful drawing skills and all. But whenever you have something in your mind, some shape or size or some characters in your mind or in your hand, then try to make it drawn on a paper with some scale added. Okay, so not only a drawing helps you to remember, but also such kind of drawing and keep updation will enhance your observation capacity also. Instead, if you go and started taking pictures and analyzing the minute characters or specific characters might not be stayed back in your mind. So the first initial step of taxonomy or field identification is drawing. And I personally don't think that something can substitute learning by drawing, because there are so many characters, so many minute differences, so many things to deal with. Okay, so drawing not only make you interested in the field, but also it helps you to go for a systematic what do you say updation. Okay, so in classic botany or in classic taxonomy, okay, botany itself, earlier days, the plants have been documented by means of such wonderful drawing, lively drawings, and so many such lively drawings, live drawings are available online. Okay, so we can just go and see and try understand. Excuse me, Naren. Sorry. Sorry. We can try understand how they have drawn these pictures, and what are all the characters that they have noted down for this particular thing. Okay, so and another feature is that this will stay for a long time. This you can deal with your children, your students, and things like that. And also, when you explain or when you introduce a new plant to the world, you can go with enough characters. But just imagine that some of the characters are hidden by some other forefront characters, then it may not be included in the, we cannot include such kind of things in the pictures, in the photographs. So you have to draw diagrams in such a way that most of the characters which are important or significant to that particular plant in the drawing. Okay, so then, I mean, we have classic examples from Hortus Malabaricus also. They've taken many days for getting such kind of illustrations in the 17th century. Okay, so now another thing is that confusing. Whenever you look at the right-hand side picture, you can easily understand that, yes, how can we distinguish these shapes? Okay, let me go in detail. So these are some of the terminologies that is used in Flora of the Presidency of Madras by J.S. Gamble. So instead of one shape like ovate, or instead of another shape like elliptic, ovate comes here, ovate is here, and elliptic is here. So when you deal these two characters, there are very minute differences. You can see that in ovate, it is the maximum width is towards the base, and in elliptic, the maximum width is at the center. So if you are getting a shape in between, like the width is little below or something like that. If the width is little above, then it would be between elliptic and ovate. And look at ovate. And if you just replace this petiole here, it will become ovate. So there are different names to categorize different shapes. So if you get such kind of explanatory terminologies, ovate, elliptic, you have to weigh and you have to try understand. And this kind of understanding will happen when you started dealing with any classic taxonomy keys or identification keys. Okay, then another thing is that when someone in the key says that slightly cordate, like this is a cordate leaf, the base is cordate, and when he or she, whoever who had written the key, I mean, we may not be able to understand what he meant by writing slightly. Okay, we don't know whether this is slightly when compared to an actual cordate, bigger cordate leaf. Okay, then there will be terms like larger, then there will be another terminologies like smaller. So we don't know how larger it can be, how smaller it can be. I mean, fortunately, if there are measurements available, but there are, along with keys, there are no, I mean, majority of the keys may not have measurements. Okay, another thing is that when you deal with the epidermal characters like hairs, we know only that there are hairs, but you look at the types of hairs described, there are many more. So whenever there are this many hairs or this many types of hairs, it is very difficult to handle. Okay, now let us see it later. Another thing is that you look at these pictures. All these pictures that I've taken from Mimosupsa elani plant, elani. a common plant. But look at the shape of the leaf. We don't know whether it is ovate. We don't know whether we should put it in elliptic, oblong, whatever it is. And look at this plant. Shape is entirely different. And many a times online sites wherein I am a moderator or I'm an expert in identification, we used to get such kind of leaves for Mimus of Selenium. But unless you have other characters like this lepidode hairs, I mean, it is very difficult. Okay, but in the same species itself, you get this kind of variation, like local variation or other variation. So in such cases, you may need to go for using ovate elliptic or elliptic oblong, oblong lanceolate, things like that. Okay, then there are lots of confusing things. Say, for example, a terminology called pubescent. Pubescent means soft hairs, but which are slightly erect. Okay, another terminology, puberulous. And you can see that they are also very soft hairs, but they are not erect at all. They may be at present, they may be attached onto the surface of the leaf. They are not at all strong. But there are other group which are the hair, wherein the hairs are very strong. That is hispid. It may pierce through your skin like a mosquito. So then hirsute, it is also erect, but it is not that strong to pierce through your skin. And there is one more thing, pylos, which is almost similar to hispid, but these are soft, erect, spreading hairs. So, I mean, unless you have this narrow distinction between these terminologies, it will be very difficult to follow keys. Okay, then similarity, another difficulty. When you look at these two plants, unfortunately, I haven't got leaf picture for this Impatiens uncinata. Okay, but let us see with the flowers. So when you look at first hand, when you look at these two flowers, you may think that these are same species. But let us see the key for these two species. One is Impatiens uncinata, another one is Impatiens campanulata. In Impatiens uncinata, not arcinata, uncinata, spur of lip short. And for those who don't know what is spur, this is the spur. This is the spur of Impatiens flower. I haven't got the spur of this particular plant or this particular flower. Then the spur will be incurved at the tip. This is the tip region, this part, and that is incurved. Then the tip is inflated. You can see that the inflated tip of the, here also, the inflated tip of the tip. But here, this plant, this particular uncinata, it is, the inflation is at the middle. The flowers are pink. And let us see the next plant. What are the spur of lip even shorter than this. So if you just see one plant and see shorter, then there is no comparative account. Then it is incurved as that of the previous one. Tip inflated. Okay, tip inflated, but the inflation is only at the tip. Okay, then flowers white with pink blotches at the throat. You can see the difference. So unless you know these characters, I mean, sometimes I used to wonder when Dr. Jomi Agustin sir tell me some of the plants which he had never seen. And I used to ask with curiosity, I used to ask him that, sir, how can you explain, how can you tell the name that you have never seen so far? And he used to say that I know the key. Whenever we go for the key, we used to, we used to start with other plants, other species also. So then you might have the key characters in your mind. So that will take you to the next plant also. Okay, then I'll tell you another species, another genus, which is Psychotria. There are so many species, but I have taken few similar, very closely related species. Psychotria of Rubiaceae family. This Psychotria truncata, Psychotria nigra, Psychotria nilgirans, Psychotria flavida. But if you go to identify these plants by using Gamble key or Gamble's flora, it is going to be really tough, because very minute characters such as hairs in the throat of the corolla and the shape of the pyrens or the seeds, planoconvex or planoconcave, things like that, number of pyrens, and calyx, whether it is micronate, sibilate, or things like that. And these characters are very minute. So we have to develop such a expertise or habit of dealing with even minute micro habit also. Then another difficulty is variation. So in different places, the same species might be appearing in different forms. So we might land up with delivering it as different species. Then ultimately we might go for even publication of species novo, and but it is nothing other than ecotypes or ecophenes because of environmental or soil, differences in soil features or climatic variation. Another thing is scientific naming. I hope that you know why scientific naming is important. Just a simple example, there are three fishes that I have shown here. The first fish is called a lionfish. Some other place it is called zebra fish. In some other place it is called firefish. In some other place it is called Turkey fish. And the second one, it is called angler fish in some place, monk fish in some other place, then French angler fish in some other place. Then the third one, blue tang, royal blue tang, hippo tang, flat-tailed surgeon fish, Pacific real blue tang, blue surgeon fish. So if someone who is speaking out one name that the other person doesn't know, then that he will be in trouble. So then it is very difficult to identify the species very precisely. Okay, so to avoid this one, we are using to make it systematic or scientific. We are using unique scientific names for different organisms, and we have to thank this particular person not for deriving this binomial nomenclature, but to popularize binomial nomenclature. It is none other than Caspar Bohine who introduced binomial nomenclature using genus and species name. Okay, and yeah, look at the interest that or the how much he dedicated himself into the field. He changed his name from Carl von Linné to Carolus Linnaeus, a Latinized name. Okay, and species plantarum is one of the pioneering work with which we've learned how to use Latin name with ease. The first part of the scientific name, which is the genus name, is a Latin noun form. And the second form is a Latin adjective form. So the Latin noun form will be the generic name or genus name. And the second form is an adjective name that usually explains specific characteristics of the organism. That is why we call that as specific epithet, which is specific, which tells you what is specific with that particular plant. Okay, so how can we remember or try remember these names? Let us try introduce 5 or 6 plants. Okay, this you might have seen this plant, Terminalia elliptica, and we know that this is crocodile bark tree because it analogous with the crocodile skin. And there are 2 terminologies. One is terminalia, and terminalia means terminally clustered. Those who remember the leaf arrangement of Terminalia elliptica don't go for this one, because the terminology has come based on Western species like Terminalia catapa and all, wherein the leaves are arranged or aggregated towards the tip. Okay, then elliptica, the leaves are elliptic. The shape of the leaf is elliptic. So if you can relate these 2 terminologies with these characters of that particular plant, then that is easy. With bark, with leaf, flowers, fruits, etc. Then another related species, Terminalia Chebula. Let us see how Terminalia Chebula evolved. Terminalia, I've told you, then Chebula means black. So, but no black things or no black structure can be seen in Chebula. Then where it comes from? So there is a dye. There is a dye which is, sometimes depending upon the method of extraction that you're using, you get a very dark black dye out of Chebula, Terminalia Chebula. And during 1900, around 1,34,000 rupees business was there in Mumbai port related to this Terminalia dye. Okay, so the name Terminalia Chebula came from the black color of the dye. And if you change the extraction method, you get different colors also. Then Terminalia paniculata, Vellamarida or Venmarida, not Lagestromia, Terminalia paniculata. Terminalia is terminally clustered. Paniculata means tufted. Tufted means, here what is tufted? You can see the tufted inflorescence, and also you can see the tufted, the fruits. It's very tightly packed, tufted features with respect to the inflorescence and the infructescence or fruits. Okay, so if relate Terminalia, terminally clustered, paniculata, which is tufted. Then another interesting terminology or name, Alstonia scolaris. The generic name came from, in commemorating a distinguished botanist called C. Alston of Edinburgh, who lived between 1685 and 1760. But the species name, specific epithet scolaris, came from this kind of a slate, a wooden slate, because the wood of this Alstonia scolaris has been used traditionally to make the frame of wooden slates. So scolaris came from this particular traditional usage, Alstonia scolaris etchipala or edilampala. Another interesting feature, this I have shared some years back in my Facebook. Look at these are fruits, and look at the heart-shaped or caudate feature on the fruit, on the seed. Actually, it is not fruit, seed. And the plant is Cardiospermum helicacabum. It would be very difficult for a new person, especially, to remember this kind of a name, Cardiospermum helicacabum. Let us see what is that. The genus name has two parts, cardio and spermum. Cardio means heart-related, or here heart-shaped. Then spermum means seed. Okay, heart-shaped seed. Then heart seed. Okay, then helicacabum means salt barrel. Look at the shape of this particular seed, and you can imagine that there are ice cream bowls also. I mean, with the lid, you can analog this particular thing to the lid of that ice cream bowl. Okay, then if you can understand and relate the terminologies, the meanings with the name of the plant, then it would be easy. Now, this is Cardiospermum helicacabum, which is a very common plant. In Malayalam, it is called Valliyuzhinya. Okay, then there are plant characteristics. These are very important, and I'm not neglecting any characters that I'm speaking out, because in some groups of plants it may be general characters that I have explained so far. In some other, whereas some other key might be handling with floral characters, where some other keys would be handling with the very minute floral or leaf characters. Okay, now for a plant, plant characteristics, when you consider the physical or basic characters such as life cycle, whether it is annuals, biennials, perennials, or semiparous, because these characters may be there in the family characters, family keys. Okay, then habit, such as trees, shrubs, herbs, vines, woody climbers or non-woody climbers, things like that. Foliage or retention capacity, deciduous or semi-evergreen or evergreen, like that. Use and location also. Geography, biogeography is very important, but although it is not mentioned in the characters, but it might be required if there is any, if there are two candidates for a particular specimen, then you have to look at whether it is distributed in Kerala or some other places only. If a plant has been recorded only from Visakhapatnam in Gamble, then there are chances are very less that the same plant is seen here. I'm not neglecting that it will not be seen in Kerala. Okay, but that is also significant and important. Then stem characters, you have to look at the texture, color, and exudates. And such things might not be there in keys. These can be, when you started learning field taxonomy, field identification, you should consider these characters also. Collect these characters, even smell also matters. I can identify certain plants by getting the smell. You just go and try with Lanea coromandalica. Lanea coromandalica, you just squash the bark and try smell it, and you just try see whether that smell is getting from any other bark or any other tree. And also you can try with Mimosopsilani, you can try with some other non-plants, and then see whether you are getting that smell from some other tree bark or not. So there are unique smells with which also you can identify. So texture is important, and color is important, color of inner bark, and exudates, resinous exudate or other color exudates or something like that. The nodal region, like whether the node is flat, swollen, stipules, whether stipule is present, whether stipules are small, I mean shape, size, things like that, whether early deciduous or caducous, whether the stipules fell off along with the spreading of leaf, or things like that. Then tender branches, whether the tender branches have any specific color, whether the tender branches or new leaves do have hairs or scales or things like that present. Okay, so look at this different plants. I have taken this from Mangalore while doing a study, and you can see that different species, these are different species. They are different. This and this may have the same coloration, but this do have exudate, which is slimy or oily. This do have the same coloration, but there is no exudate. This do have exudate, but it is not oily. Okay, here also the pattern is very different. Here also green inner bark with an inner slightly pinkish or reddish coloration. Okay, so such are the characters which would help you for field identification. You may not be getting keys, but some experts might be asking you what was the color of the bark or the inner bark. Then plant part, you should know these structures, and I mean precisely what are the differences between thorns, spines, prickles. Let us see what are these, what are the differences. A thorn is a modified stem which is subtended by leaves at the node, which may have a very sharp tip. Okay, whereas a spine is a modified leaf or stipule which is having a very sharp pointed point at the tip. Then it might be emerging sometimes from a leaf. Okay, Acanthus illicifolius, a mangrove plant, if you look at, means you may not be able to collect the leaf because of the presence of this kind of spines along the margin. Okay, so those are spines, those are not thorns. So when you get spines in a key, then you have to relate it with a modification of leaf or stipule. And whenever you see thorn, then don't confuse it with the spines. And let us see what is a prickle. Comparable to hairs, because they are not neither modification of stem nor stipule or leaf. They are emerging from the cortex of or epidermis like that of hairs. Okay, and it may be born in places other than nodal region, like that of this particular one there in Rosa, wild rose, this one. Okay, so such kind of minute details or minute definitions you might be, you should be familiar with. Then you should, when you go to leaf, there comes lots of tricky parts, lots of difficult parts. Arrangement of leaf that we all are familiar with. Then shapes of leaf, which is tougher. Color of leaf, but beware of change in color at different places. Deciduous, the same species may be keeping a pattern of coloration than when compared to the same species in evergreen. Okay, then pattern of venation. This may be consistent across species. There won't be much changes. So, okay, symbol to compound, that comes in arrangement. Okay, then margin, there also, of which of these shape, margin and pattern of venation is very important. The French Research Institute people, they came up with a key for forest trees and lianas of evergreen plants of the Western Ghats. And I have utilized that extensively during my PhD work, because they have mostly depended on leaf characters, because the title itself is leaf key. Okay, so venation pattern, shape and margin was the main characteristic features that they have used for deriving key. They have derived a key also. The book is available with the French Research Institute, which is very nicely done. Okay, then the surface, the texture, the presence or absence of hairs and types of hairs. Leaf arrangement, we know these leaf arrangement, alternate, opposite, decussate, then spiral arranged leaf. This is also a spirally arranged leaf. Then a whorled arrangement of leaf. Then compound leaf, whether it is palmately compound like this, then pinnately compound. If it is pinnately compound, it is imparipinnate or paripinnately compound, and a number of leaflets, their shape, things like that. Then bipinnately compound, tripinnately compound leaves. These are the characters that would be determining family or genus level. species level, we may not be able to depend leaf arrangements like this. But when you come to the shape of the leaf, it is very crucial in running the key of flora of presidency of Madras, because several species have been handled by using leaves. Because many a time Gamble used to collect plants, or Gamble's team used to collect plants, and whichever which was not named or no name available, he used to send it to Q, and there, if there is no flower, and they will be deriving key based on the available leaf or leafy twig. So therefore we have differences in keys of Gamble's flora. Many of the keys are dealt with only leaf characters. There are certain plants which possess scale-like leaves or all-like leaves. All means the needle-like tip or needle-like character, which you are seeing in a spikelet or in a gramineous flower. Or sometimes you may get needle-like leaves like that of Casuarina, or not Casuarina, sorry, like an orchid called Lucia abrahami, Lucia macaranda, evangeline, things like that. Okay, then leaf margin. We might not have noticed this many different types of leaf margins. There are different names also for different leaf margins. I'm not trying to explain each and every character, because I don't think that this 1 hour would be enough for explaining these kind of characters. But I'm trying to say, trying to explain what are all the things that you should be taken care of. Okay, so there are so many different explanations with respect to differences in the leaf margin. Okay, so shape, leaf margin. Then the tip of the leaf. What are the pattern of the apical region of the leaf? Whether it is acute, whether it is apiculate, aristate, cordate, then emarginate, mucronate, obtuse, retuse, things like that. The venation is also there, and also base, whether it is auriculate, cordate, feltate. There are so many different explanations. So unless you know these terminologies, you may not be able to run a leaf key character. Again, venation pattern. And from species to species, there may be differences. When you take a closely related genera, you may be able to classify by looking at the venation pattern of venation of a typical leaf. Okay, so there may be pinnately arranged veins within a leaf, and we know that there are parallel or reticulated or netted venation among angiosperms, parallel venation in monocots and reticulated venation in dicots. Some of the monocots also do possess reticulated venation like smilax. Okay, but pinnate venation, like the primary veins are arranged pinnately like this. Then acute venation, this is a very different venation. Then palmately veined, at least at the base. If you can see, the primary veins are arising as if they are, as if it is like a palmate leaves, then you can call this as palmate venation. And there are n number of types of venation, but I mean, they are not classified. You can rely upon venation for identifying or distinguishing plants in the field. Okay, then this is a reticulated venation with pinnately arranged primary nerves or veins. This is a palmately arranged leaf of a Dioscorea. Excuse me. A parallel venation in banana leaf. And this is a wonderful picture that I've got from internet. Parallel venation. Parallel venation in monocot leaf. Okay, then leaf surface. And we've seen this picture earlier. There was another picture also. But there are few characters which we can consider as common characters. One is glabrous without any hairs. If a leaf doesn't have any hairs that you can consider as glabrous, then there are so many different characters or names or terminologies for differences in characters. For this, you have to have a very good hand lens or a microscope, and I'll tell you how to look at it. It's not like taking sections and look through microscope. I'll tell you, I'll show you how to look through this, look for this particular character. But you have to have a keen eye, keen observation to discriminate or to differentiate which is this one, whether it is glandula or lanate or hirsute or hirpelose or hispid, things like that. Okay, and also you have to look at a new leaf. This I've already shown you, but again I'm showing it here because to support whatever I've explained a little before. Okay, then leaf surface, how we can look at it. There is one more, there are two more. This is tomentose. This particular character is called tomentose, covered by tomentum, covered with, sometimes it may be covered with slight hairs also. So then you then the key or the person might be calling this as tomentose to pubescent. Okay, so this do have tomentose nature, this particular one, and also it do have soft hairs. Okay, then scabrous. Scabrous leaf surfaces having hairs which are very strong, very short and very strong prickly hairs. It might pierce through. An example is Ficus exasperata. Theragam ennu vilikkunna oru plant, Ficus. Earlier people used to use that as, in place of sandpaper. People used to use that for cleaning woody furnitures and woody structures. Okay, so this could be the microscopic picture of, or a macro picture of scabrous, very pointed, strong teeth-like structure. Okay, and how can we look at it? You can even in the field, if you hold the leaf like this, you just hold it downward one side and see whether there are any characters like this. And this is the best way to go about in looking at. But if it is a stem, then you need to take some part detached from the plant and then observe it under a microscope, or take pictures and zoom it and see, or whatever which is convenient. And then this is a very different condition, which is glabrous condition without any hairs. And among glabrous itself, shining, then waxy or oily with oil glands. There are so many different types of glabrous leaves are there, but no hairs. Okay, then these leaves are glabrous, but when you take the underside, you can see a kind of silvery or very whitish coloration. And this particular condition is called glaucous, especially glaucous beneath. Sometimes the leaf upper surface is also slightly glaucous. Okay, so this might be presence of wax coating above the leaf. Another condition is rugose. I don't know whether you have seen this type of plant. So this is a particular endemic plant seen in the evergreen forests of Western Ghats, which is in Malayalam, I think it is called Aryapurian. Okay, the surface is ringly like this. This plant is Kunstlera carolensis, which is very common in Kerala, Tamil Nadu, and Karnataka. Okay, then glandula. Some of the leaves, even when you touch it, you can easily understand that glandular leaves. Not only leaves, but also the pedicel, peduncle, or even some of the internodal region also, stipule or whatever it is. So there are so many glandular structures, and these glands might be secreting. Secretory glands are there, non-secretory glands are also there, and this might be secreting different exudates. So depending upon the nature, depending upon the structure, you can classify glands. Then gland-dotted leaves. The glands are embedded in the leaf. So when you keep the leaf against light, you can see like this, that there are glands. Not only the leaves, but also the veins are also gland-dotted. So such leaves are called gland-dotted leaves, especially in the family Rubiaceae. Okay, then when you go to plant flowers, which is a little more tougher. But I would suggest you don't go for color, shape, and size. Size, obviously. Color is not a very typical or good character for distinguishing species, because there may be changes in color depending upon light, depending upon even nutrients, depending upon other climatic factors also. You can easily, even if you travel from Cochin to Munnar, you can easily see the difference in rosa flower itself. The color that you get in Munnar may not be seen in Kochi. Okay, so then flower is very typical for angiosperms, and there are so many characters that we can look at. I'm not explaining these characters because assume that you are familiar with these terminologies. So we have to look at some basic classifications, broad level classifications like actinomorphic flowers, zygomorphic flowers, then bisexual, unisexual, and also when you get one sex, the flower of one sex of a unisexual flower, then you have to try get the other sex also, the flowers of the other sex also, because even leaf characters or stem characters might also vary in both the sexes. Okay, then when you, I mean, this is going to be tougher. Eclamatous flowers and very microscopic flowers like this one. This is a Podostomaceae plant, and it may come out of the water surface only during summer season. And during monsoon or when water is there, and when you see the stem of this particular plant, you might be thinking that this might be a bryophyte plant which is not yet described. Okay, and there are flowers like this also, and there are petals. There are sepals, and when you take sepals of different flowers, I mean, you can see the variation in arrangement, in shape, size, in venation of sepals. Here also venation, and also presence or absence of hairs or pubescence or whatever it is. Okay, in different family, you may get different types of sepals, and these all are polypetalae. I mean, comes under polypetalae. And when you come to gamopetalae, and one major thing that you can notice is that most of the gamopetalous sepals are also gamosepalous. Here also you get gamosepalous sepals or calyx. But most of the polysepalous sepals you get in polypetalae. Okay, here most of, and also mostly non-hairy. But in some families like Lamiaceae, Scrophulariaceae, you get hairy calyx. Otherwise, most of the gamopetalae sepals are non-hairy. Okay, then petals also. Polypetalous petals, I mean, they are polypetalous. But you can identify if you see a petaloid flower from a polypetalous group, then you can easily reach a family or a family near to the specific family. Of which one thing that I want to specifically mention, this belonging to the family Myrtaceae. And the petal, I mean, the typical petal is absent from this, but instead there is a cup-like structure, and you can see that it is a union. And this cup-like structure is called the Calyptra. And those flowers that possess this calyptrate structure is called calyptrate. The flowers are calyptrate. And the size, shape, and nature of, and this may shed off once the flower opens, because the stamens pushes the calyptrate cap-like structure and they may fall down. And the remaining part of the petal would stay back along with the, but this would be mostly considered as sepals. Okay, then there are gamopetalous condition, but I have placed this also gamopetalous. Some people, and this also. When you see these two, you may confuse that, is it gamopetalae? Yes, they are gamopetalae, but their fusion is only at the base. This forms a tubular corolla, and here it may fuse to form a cup-like structure at the base with adjacent to the nectary. Okay, so you may be able to distinguish characters specific to a family, like the general features of petals or sepals. Then there are characters which is related to anther and anthers, like monothecous anther, bithecous anther, and also the nature of union of anther, Delphus condition, or it is androus condition. Then what else? The attachment of the stalk onto the stamens, things like that. Gynoecium also contribute to several characters, like whether the carpels are free or united, apocarpus or syncarpus, then whether there are hairs associated with carpels, and how they are arranged, and whether there is any other structures associated with ovary or carpel, then presence or absence of style and length of style, nature of style, then stigma, things like that, then a number of ovules and all those things. Okay, let me get into a classification and how to deal with classification. So Bentham and Hooker's classification is the one that we are following here. And nowadays, I don't know, people tend to produce flora based on APG system of classification. But to me, I personally do not agree with the publication of flora based on APG system of classification, because that is not usable in the field. We cannot identify plants by running a key using an APG system of classification, because they haven't taken much of the field characters. Okay, so Bentham and Hooker's classification is very unique in such a way that they have considered lots of such features. And this is the most important flora that we are following, and we have to thank J.S. Gamble, who dies off immediately after the second volume is completed, and Fisher and Mrs. Gamble completed the third volume. And there is a huge difference between first, second, and third volume. Third volume from first and second. Okay, so this is the general classification of Bentham and Hooker. And when you take Gamble's flora or flora of Presidency of Madras, you can see this this kind of an arrangement inside. There is a very well prepared key, and unfortunately there are no pictures. Unfortunately, there are no diagrams. But I strongly suggest this as the best key, at least for some families, several families, because your imagination and observation will go up. And also you tend to collect plants, you tend to draw diagrams, you tend to collect pictures. Okay, so this is a very good flora or key for starting practicing. Okay, then once you handled this key, then there won't be any problem in handling other keys. Okay, it is just like if you can ride a bike or a car inside the city, then you don't feel any discomfort in riding bikes or cars in outer skirts. Okay, so now let us see how the keys are developed. This is the basic key going into the family or subclass or series. There are Polypetalus flowers. They comes under Polypetalae with free petals and stamens free from petals. Then Gomopetalus flowers, wherein the petals are united and stamens are born on the petals. And then there are Monoclamidae, wherein only one clamid is present. This is a Litzia species of Lauraceae family, wherein only one perianth whorl is present, Monoclamidae. Then we go to Inferae, wherein inferior ovary and flowers are epigynous, stamens equaling to the number of petals. Then Heteromerae, wherein the flowers are hypogynous, stamens as many or double the number of petals. Then Bicarpellate, wherein the number of carpels are restricted to 2. Then stamens as many as that of petals, and they are alternating. Okay. So such is the key, and you can dissect a flower, and you can straight away get into such kind of broader level of classification. This is for a beginner, for an MSc student to start doing. Okay, then identification of family. Then there are so many general characters for identification of a family. An expert can easily say, even by looking at the leaf itself, or by looking at the flower itself, that this might be belonging to XYZ family, or something which is related to that. So there are different such kind of system, and I suggest each and everyone to personalize such kind of characters and to go for identification. There are certain keys, certain information available online also, but these are generated out of experience. And so like this, flowers also. This, even for a BSc student who learned taxonomy in BSc level, can easily say that these are capitulum inflorescens belonging to the family Asteraceae. There won't be any doubt. There are certain other characters that you can depend upon straight away going to the family. So if you get a Rutaceae plant, You can use, I mean, one thing that you can smell, or you can look at the what you say, polyadelphous condition, or Obdiplostomonous condition, or the petals nature, or things like that. Likewise, Lamiaceae, we have characters. Okay, then these plants belonging to the family Rubiaceae. If you see opposite decussate leaves, then what is inter or intrapetiolar stipule, then you can straight away go to Rubiaceae family. Okay, so then identification of genera. Once you identify a family, then the next problem is identifying a genera. At this level, you may require to go for genera unless you know the genera earlier. Okay, the genus earlier. Okay, so I have some plants here, and some of you might know that this belongs to the families as Alpiniaceae, and but it is very tough. From each and every photographs belongs to one species each. Okay, so it would be very difficult. So we have reached the conclusion that it is the Alpiniaceae family. So then you get into gamble, and then you have key like this. Now you run the key, leaves all bipinnate. We have no leaves which are bipinnate. Okay, so the tribe, this particular tribe can be excluded. No other tribes are there. I go to the next tribe, Cassia. Leaves simply pinnate. Yes, calyx lobes usually free to the base. I mean, look at the calyx base and see whether it is true or not. Okay, leaves of 2 leaflets, no. Leaves usually abruptly pinnate, no. So I stick on to Cassia, and then petals 5, here petals 0. Okay, then I can easily go to the genus Cassia. Petals 5, you cannot, you don't even, because only 2 genera are there. One is Cassia and the other one is Dalium. And now I've reached the genus. The new name of Cassia is Senna. So Senna or Cassia. And now you have to use the species key. Using a species key is laborious. You may need to spend time. Whenever I started identifying plants, I used to spend hours together or sometimes days together. I took 3 or 4 days to identify. Sometimes you may not have some certain characters, then you have to go back to the field, which may be far away inside the forest. So then you have to collect other information and come back and then again run the key. Then you would be fed up with running the key, and then you have to leave some time for a change and then go back to the thing. So there may be so many hurdles. Okay, when you started running the key, this is a very different key. This I've taken from eFlora of China. So petiole and rachis of leaves without glands. In Cassia, if you go for a close observation, there are glands present along the rachis, or at the base of the rachis, or at the base of the petiole of each leaflets. So here it says that petiole and rachis of leaves without glands. If that is so, then go to 2. This is 2, and there is only one species, Senna alata, and this is Senna alata or Cassia alata. This is very familiar to you, I guess. Okay. Then this we have identified. No, it is not going to. I mean, there are what you say, glands available. So then petiole and rachis of leaves with 2 longitudinal ribs. Along the ribs, there are 2 longitudinal what do you say, upright portion, which is narrowly winged. Legume or the fruit with a membranous wing at the middle downward to the base of each valve. You have to split open and see. Then if this is agreeable, then go to 5. In between, if you are not ending up with this Senna alata, then you have to go for the additional key for 2. Petiole and rachis of leaves without wings. Okay. Then you have to go to 3. Okay. Then 3 deals with leaflets abaxially densely yellowish brown velutinous, legumes narrowly cylindric, and this is spectabilis. This is very common in Bangalore and other places, which the fruits looks exactly like that of Cassia fistula, and you can see one fruit here. I'm sorry, I haven't taken fruit pictures. Okay, this is Senna spectabilis. Okay, then okay, if you are not getting, so you are here at 4, you are not getting this particular key, then uh, read under under 3 plus. Leaflets abaxially glabrous. That means there is no yellowish brown velutinous, which is very minute. We may not be able to see yellowish brown from a distance. You may be able to see it is a fine structure. Okay, and which may be so. It is glabrous, and sometimes it may be uh, farina white, slightly or slightly whitish, legume flattened. If this is the one, then of the plan that you have got, then go to 4. And 4 deals with 2 plants, leaflets 6 to 10 pairs. Okay, Cassia Siamia, leaflets 6 to 10 pairs. Okay, then what is the other one? Leaflets 8 to 16 pairs. When some people may ask that, sir, this 10 is also included here, then how can it be 9 and 10 will, if I get a 9 and 10 and it may not, if it is not Senna Siamia, then what will I do? So since this variation is there, it can be any number between these two. But what you have to do is that you have to collect as many number of leaflets, as many number of leaves, and see what is the typical number of leaflets, an average number of leaflets in that particular species and that particular individual. Okay, then stipules ovate chordate, and this one, the stipule was linear and caducous. Then tardially deciduous. മിക്കവാറും അത് എന്താണ് പറയുക, ഫ്ലവർ വിരിഞ്ഞു തുടങ്ങുന്ന സമയത്ത് തന്നെ പൊഴിഞ്ഞു പോകും. Early caducous or early deciduous. Didymobotria. Let us see what is that. You can see that the number of leaflets here and also the size of the leaflets. But when you derive general key for the genus, it may be
ഹലോ ഹലോ ഹലോ
ഹലോ സാറേ കേൾക്കാം. ഹലോ കേൾക്കാം സാറേ.
കേൾക്കാം. എനിക്ക് തിരിച്ചു കേൾക്കുന്നത് അത്ര ക്ലിയർ അല്ല. എനിവേ, ഓക്കെ ഓക്കെ, ഐ എം സോറി ഫോർ ദി പ്രോബ്ലം. ലെറ്റ് മീ okay, can you see my screen? Can you see my screen?
Yes, sir.
Okay, okay, okay. So the only problem is that I am now operating from another computer, so I may not be able to show you pictures of these key. Okay, so I hope that you could understand how to deal with a key. So you are, I mean, in such cases, the confusion that we were discussing earlier might arise. So in such cases, what you should do is that you have to discriminate or given weightage to characters which is most prominent or most common in that particular individual or group of individual you believe comes under the same morpho species. Okay, so such is the way to discriminate one species to the other. That sometimes you may be able to do that in the field. Sometimes you have to take a multiple number of specimens or branches or whatever, and then come back to the lab or your workplace, and then identify and started running the key. Okay, so I'm sorry that let me just check whether it works. No, it is not, because the folder has changed. Okay, so then yeah, this is one key for what do you say, for explaining one genera. And there are so many other keys available, but this is little easy when compared to some very confusing or very what do you say, tough key such as Crotalaria. The key for Crotalaria in Gamble is very difficult, and also the key for impatience in Gamble is very difficult. And there are so many other genera in Gamble, it is very difficult. But take it as a challenge, and if you can identify some of such challenging genera, then you can go for any other easy or any other even tougher ones. Okay, so now there are other, I mean, nowadays it is there are lots of online applications, there are lots of online keys available. Now when you go to Kew Botanical Garden site, you may see flora of Sambesica. I don't know which place is this, but they have, I mean, somebody have done the flora of this particular place, as I think it is an African place, and then they have what is it, come up with a key. And then you can go to the plant list run by several botanical institutions, and then you can enter a particular plant's name here in the search region and search for identifying or understanding the new name or change in names or synonyms and things like that. So you can authenticate your identification, and you can go for the identification with the help of the plantlist.org. And also there is another thing which is initiated in India, run by or initiated by an engineer called J.M. Garg. This is eFlora of India, and I'm fortunate that I am one of the moderators in this particular online group. So this is a very good group wherein you can send your pictures, and there are lots of experts across the country, and there are some experts who are outside the country, and there will be a discussion forum, and there will be discussion by different experts, and then they will be coming to a conclusion. Okay, and in due course they will be asking for more information such as place, date, flower, I mean color, if the color is not very clear from the picture, and other information if required. Okay, then eFlora of India. Now, and also you can go to the site and then you can rightly enter into the families, and then when you click the family, then you get this list of genus. When you click each and every genus, you go to the page wherein the species of each genera is kept. Okay, teacher. Yo. Okay, ipo clear ആയില്ലേ? ലിങ്ക് ക്ലിക്ക് ചെയ്തപ്പോൾ അത് നെറ്റിലേക്ക് കയറിയതാണ്. Okay, thank you. Okay, so when you go this particular eFlora of India, there are family names, and then below, I mean, under each and every family, there are names of genera. When you click each and every genera, you can see species under that particular, and also you can see discussion in that particular thread. Okay, discussion be different people. This comes under blacklist. So there are different species and pictures of different people from across the country, so that you can see differences, variations, and things like that. And there are sites in Facebook, Flora of India, Flora of the Western Ghats, Flora of South India, Flora of so many, garden, so many different sites are there, and groups are there in Facebook also. Okay, so I'm stopping here, and I think it is just a beginning of the journey. So I hope you got some interesting aspects, and I'm sorry for the interruption happened in between. Okay, and luckily there was no power interruption, because if power interruption happened, means I would have been in darkness, and it would have been little more troublesome. Okay, I would like to thank Mrs. Mariama teacher, my teacher who brought me up as a botanist and a taxonomist, who taught me taxonomy. Was a retired professor from MA College Botany Department, and now residing at Alua. And also, obviously, Dr. Jomi Agustin, who taught me the field taxonomy or field identification of plants, several information or knowledge which is not available in taxonomy books or flora books. So thank you, sir. And also my college. Thank you.
Thank you, sir, for this wonderful presentation. In fact, it was a very informative. And if anyone has to ask something, you can post your questions in the chat box.
ടീച്ചറെ എനിക്ക് ശരിക്കും കൂടെ കേൾക്കണില്ല. ഞാൻ നോക്കട്ടെ കേട്ടോ, ഇവിടെ കുറച്ചിട്ട് പോകണം. ഒരു പണി, ചാറ്റിൽ കൂടി ഇതുണ്ടെങ്കിലേ, ക്വസ്റ്റ്യൻസ് ഉണ്ടെങ്കിൽ ചാറ്റിലും കൂടി എഴുതാം. ഓക്കെ. Shebin, there is a question. There is an interesting question. Cisalpinaceae family, both the Hypogynous and Perigynous are seen. Can you give example? Is Perigynous is evolved from? Oh, that is little tough. The question is little tough, but the aspect that the question deals with very interesting, because I mean, so many people are still in debate that whether it is Hypogynous or Perigynous. But when you consider the ovary is superior. But when we teach taxonomy or morphology, we call that as a perigynous flower. Okay, but it is not actually arising from middle of the ovary. It is independently starting from below the ovary. So technically, it is actually a hypogynous flower. It is not actually this thing. What do you say? And even the stalk of the ovary, that is arising from below the what do you say, the other floral walls, the hypanthial cup. Hypanthial cup is none other than calyx. But unfortunately, we don't have any other family or any other plant to explain perigynous flower. So we are still keeping that as perigynous. Okay, but evolution to explain the evolutionary aspect of perigyny, epigyny, and things like that, it would be a little difficult. I think if you can send me your email ID, we can discuss our email. It is little explanatory.
Sir, there is another question. For grass identification, which flora will be more good?
Okay, that's also good. And nowadays, if someone is very interested in the grass taxonomy, that is very cool. Okay, there are few books. Grasses of India, Pakistan, Burma, Bangladesh is available. There is one book by BSI. Grasses of India, Burma, Bangladesh, even Pakistan, all other countries are also included, I guess. But that is very tough, because key is very tough. And there are grasses of Kerala. There are two books by grasses of Kerala, I guess, and few other information also. And there are very few experts on grasses, like PV Sri Kumar sir and all. So if you can consult with such experts, because these people, they have developed such expertise by their effort. It is not by looking at this thing. And also herbarium.
Then another person whom I what do you say recommend is Dr. A.K. Pradeep in Calicut University. He is good at grass taxonomy. And Jomi sir is also, obviously, he is also a very good grass taxonomist. But the only thing is that these people have been specialized in grasses, but they know how to deal with the grasses. So grass taxonomy is very different. And also, I mean, if you take different groups like orchids, like Rubiaceae, Fabaceae, there are differences in dealing these different families. So yeah, I mean, some specific books I can send you as a reference. I mean, including titles and all. I request the organizers to collect email IDs of those who are interested in getting such information.
Sir, is there any method to dissect tiny flowers?
You have to have a very good microscope, one thing. And the other thing is that you should have enough patience, primarily. And another thing that you should be dedicated in sitting with the microscope and these technical aspects with pencil, flowers, and all for a long time. So another thing is that you have to have a background information about what are all the tiny structures which might be present in this in a particular plant in your hand. Okay, because in some plants the tiny structures may be the ovaries or the attachment of the placenta or or some other minute hairs or something like that, or some other time it may be the minute characters are on the calyx, hairy calyx, subulate, mucronate, or some such kind of what do you say, this thing. So all you require is that have a very good hand lens or a powerful 20x, at least 20x what do you say, dissection microscope lens. Then the recent trend is that if you can have a good camera, don't go for a highly expensive DSLR camera and all. I would suggest you go for a compact lens camera. I mean, a normal camera, preferably Lumix. Lumix is little expensive, but Lumix is the best one for micro photography. So take micro pictures and then what do you say, high resolution micro pictures, and then expand it. But remember one thing, you may not have scale for such pictures which you are going to enlarge.
So for aquatic plants, herbarium preparation, any easy methods?
Sorry.
For the preparation of herbarium of aquatic plants, is there any easy methods? Because it is very difficult.
Aquatic plants, I mean, see, the herbarium preparation is altogether a very different thing. Not only aquatic plants. I mean, are you asking herbarium or some other?
Herbarium of aquatic plants.
Okay, so not only aquatic plants, some xerophytic plants, impatiens. There are so many plants which are very difficult. So what you should do is that when you collect, you have to let the water content goes off. So for that you can put it in a I mean, among paper without applying any weight. Okay, don't apply any weight when you want to get the plant without any water. So the first day you keep it in a paper, and remember one thing, that you may not be able to detach the plant after it dries off. So in the first day, after the first day, then it may be 50% water would have gone. Then what you should do is that you get a fresh paper and then put it from up, and then keep it like this, and then turn it around. Okay, and turn it around, and the older paper, you remove the older paper very slowly, but keep something which is longer, like a needle, a long forceps or a long twig, and then or a scale, then you just try to remove the plant from the older paper slowly, and then, I mean, it takes a long time. Okay, and then there are so many other methods are also there for treating what do you say, the xerophytic plants and such similar plants. There is a question, can you send key of Orchidaceae? That is very tough because Orchidaceae, there are keys, but I don't think that there is a specific key for all the Orchidaceae members. So what you can do is that I would suggest one book, but there are lots of mistakes and there are lots of updations happened in that book. An Introduction to the Orchids of Kerala by Abraham and Valsala. Then another book is The Orchids of Nilgiris by J. Joseph. So these two books are the one that I have started using for identification of orchids, but you have to use some other methods also for identification of orchids, because key characters are very tough. And nowadays there is a new book of orchids of India by different authors. One of my friend Jivan Singh Jalal is also an author. So I haven't got that book. There he was. He had sent me 2 or 3 keys for. I want to deliver a talk on Orchidaceae tomorrow, so I got a key from him, and which is a little easy, comparatively easier than that of the previous ones. If anybody want to continue discuss, or if anybody want to do more clarification, you can write to the organizers, or you can write to me directly. If you write to the organizers, they will redirect the mail or whatever to me, so that I can communicate with you. Thank you very much for such a wonderful participation, and thank you very much for the organizers, Ajit teacher, and the institute for such a wonderful opportunity. Thank you.